Hello everyone, and welcome to another recorded lecture of BIO101 online. Today I'll be discussing Chapter 5, which is Membranes and Cell Transport. So, if you recall from Chapter 4, we introduced the structure and function of the plasma membrane. And plasma membranes are found on all types of cells. So whether you are a bacteria cell, a fungus, an animal cell, plant cell, you have a plasma membrane surrounding your cytoplasm. And this plasma membrane is referred to as a fluid mosaic. It's a fluid because it's always moving around. It's not like a solid that stays in place. And it's a mosaic because it's consisting of lots of smaller parts all put together as a larger whole. So let's take a look at those smaller parts that make up the plasma membrane. The main component is the phospholipid part. So we know what a phospholipid is. We introduced that very early on. Phospholipids have a hydrophilic head and a hydrophobic or two hydrophobic tails. And they make up a bilayer where the tails associate with each other, making this hydrophobic region. And the polar heads face the inside, which is the water on the inside of the cell, the cytoplasm, and the other heads face the water on the outside. So that's the phospholipid bilayer, and we have hydrophobic and hydrophilic regions. Phospholipids are called amphipathic. So that means that they have both hydrophobic and hydrophilic portions. So they're not strictly hydrophobic, they're not strictly hydrophilic, right? They have a little bit of both. So they're amphipathic. Membrane proteins. This is an example of a membrane protein here in orange. They can be either on the periphery, so like meaning on the inside of the cell. So they can be peripheral or they can be integral. So this is an integral membrane protein because it's integrated inside of the membrane. Whereas a peripheral protein is found on the inner surfaces. So an integral protein like this, those are like the olives in the example I gave of the bread and butter sandwich last time. So these integral proteins are transmembrane proteins because they span both sides of the membrane. And these are really important for many things, as we'll see in a slide or two. Finally, we have cholesterol. And that's what's also inside the membrane. It's inside the bilayer, and that helps with the fluidity. It makes sure that at high temperatures, the membrane isn't too fluid, and at cold temperatures, it's not too solid. So cholesterol helps regulate the consistency and the fluidity of the membrane. And I'll also now show you a little bit about how lipids and proteins may be glycosylated, which means they might have sugars added to them. So in glycosylation, carbohydrates contribute to the cell's fingerprint. So each cell in your body is your cell because it has a specific pattern of sugars on the membrane. This is called glycosylation. Think about glucose in glycosylation. So all of the cells that make you up have a specific pattern of sugars on their membrane. And this is why there's tissue transplant rejection. So sometimes if a patient receives somebody else's blood, let's say, let's take an example of that, not even tissue, let's take an example of blood donation. So what makes if you're type A positive, for example, what makes you A positive is a certain type of sugar on your red blood cells. What makes somebody B positive is a different type of sugar. If you were to mix A type blood and B type blood, the immune system would have a problem with that, and it would attack the foreign blood as being an invader. So glycosylation helps the body know who's who, so to speak, and what's what. So the sugar could be added to proteins. So the proteins on the membrane could be attached with carbohydrate chains. So these can be sugars that are added to the membrane proteins. Or the lipids of the membrane can also be glycosylated. So the phospholipids themselves can have carbohydrates, or the protein can have carbohydrates. And again, these are usually sugars. And these sugars contribute to the cell's identity. And the carbohydrate chains only exist on the outside of the membrane. So this is what makes the membrane asymmetrical, right? The outside is not like the inside exactly. And the phospholipids themselves can actually flip places. So they can go from inside and out. It's not so common. And there are certain enzymes called flipases that actually help these phospholipids flip around if needed. So here's a nice image of the fluid mosaic model. Primarily, we have the phospholipid bilayer. Here we have some integral proteins, integral membrane proteins. Here's a peripheral membrane protein. Here we can see some cholesterol. Inside of the cell is the cytoplasm. And sometimes the cytoskeleton helps anchor these integral proteins into place. So these are anchored in part of this, just part of the cytoskeleton on the inside of the cell. On the outside of the cell, you can see some of the phospholipids have these sugars attached to them, right? This is a glycolipid. This lipid has been glycosylated. And here you have a glycoprotein. Here's a protein with some sugars attached. So this is a nice illustration of this mosaic, right? Lots of different things going on. And remember, this is outside of every single one of your trillions and trillions of cells right now. So now let's watch a quick video that summarizes the lipid bilayer.
These materials move in and out of cells by passing through the plasma membrane of the cell. The plasma membrane surrounds the cell and separates the interior of the cell from its environment. This semi-permeable membrane is composed of a lipid bilayer. Phospholipids make up a large part of the membrane and form a bilayer. The structure of the bilayer is due to the tail-to-tail packing of the nonpolar hydrophobic tails, composed of two fatty acid chains, and the polar hydrophilic heads, composed of glycerol and phosphorylated alcohol. The lipid bilayer is 5 to 10 nanometers thick and is embedded with proteins. Some cell membranes also contain cholesterol. A plasma membrane contains different types of proteins which are specific to the particular function of the cell. These proteins also enable the cell to interact with its environment. The entire structure of the plasma membrane can be described as a fluid mosaic model. The phospholipid bilayer has properties resembling fluids, and the differing proteins and their attachments on either side of the membrane resemble a mosaic.
So, like that video just said, membrane proteins help give each cell an identity, and the membrane proteins do a lot of the work that a cell needs to understand what's going on the outside. So I gave the example of the membrane being like the brain of a cell, because there are all these different receptors and all these different channels on the outside of every single one of the cells that helps the cell interact with the outside world. And also helps the cell know what's going on and what's needed at any time. So we're not going to talk about too much detail, but I want you to be familiar with some types of membrane proteins. So one we spoke about already are like glycoproteins, like the MHC, the major histocompatibility complex that's on your cells that make you you. So these are the identity proteins, like MHC. There are other proteins that help transport ions in and out. We'll discuss those today. There are some that, again, allow other molecules in and out. There are some that can help catalyze a reaction. They can help speed up a certain reaction on the inside of the cell. They can also help cells stick together. We'll talk about those as well today. And as I just want to show you briefly, some membranes are called receptors. So these are like the brain proteins that are on the membrane, and they're waiting for a signal on the outside of the cell to interact with them. And once this specific signal binds to this receptor, something will happen on the inside of the cell. So that's how the cell knows what's going on the outside. So just again, very brief, basic example. As an egg gets fertilized and then divides into an embryo and then finally a newborn, cells have to talk to each other, right? They have to tell each other what to do, what has to go where, and what has to be done at what time. So in order for cells to communicate with each other, they rely on signaling molecules. And again, this is a very brief overview. I'll talk a little bit more about this in a later chapter. But the first step is that a receptor protein on the membrane binds to a signaling molecule. And that would activate this receptor, just like a lock and key. So this gets activated. And then on the inside of the cell, there's what's called a signal transduction pathway. It's a series of one protein activating another, and finally there's some kind of response inside of the cell. So there's a target protein that will bring about a change inside the cell in direct response to whatever this signaling molecule is. So those responses can be a change in the cell's shape or movement, so it can tell the cell to move or change its shape. It could be the activation of an enzyme, which can change the function of the cell. Or it can be a regulatory protein that changes gene expression. So it can change the amount of proteins that are being made from different gene transcripts. So that's a brief overview of one type of membrane protein, which is the receptor protein. So back to the plasma membrane. We said the plasma membrane is a selectively permeable barrier, right? That doesn't mean anything can get through. It's selective about what can get through. And solutes, right, these substances can enter and exit cells by different methods. We'll talk about these different methods. But the method depends on two factors, really. It depends on how much of the substance there is inside versus outside of the cell. That will determine the type of transport. And also the chemical nature of the substance. Is it small? Is it polar? Is it large? Right, that will determine how it can enter the cell. So depending on how much of it there is and what it is exactly will determine how it can get in or out of the cell. So what is the membrane impermeable to? So that means what cannot cross. So take a second to think about what cannot cross the membrane on its own. Water and other polar molecules cannot cross the membrane on their own. Membrane proteins called aquaporins actually allow water in and out of the cell membrane. And other polar molecules like amino acids and sugars need to go through membrane channel proteins in order to get inside of the cell. Ions, which are charged atoms, need to be assisted by carrier proteins in the cell. So ions, which are charged, cannot get through the membrane by themselves either. And if you're a large molecule, unfortunately, you cannot just diffuse through this bilayer, right? You need to enter or exit the cell via bulk transport in vesicles. And we'll talk about that last. So just to summarize, here we have our phospholipid bilayer. The nonpolar hydrophobic core is on the inside. And any charged molecule or ion cannot enter or exit the cell on its own. Water has to go through a special channel protein called an aquaporin. A non-charged, nonpolar molecule is the only thing that can easily cross the membrane. Small, nonpolar, hydrophobic molecules like steroids, some vitamins, some hormones can cross the membrane on their own. Right, large macromolecules like proteins cannot cross the membrane on their own. So make sure you understand this at this point. So now let's talk about the concentration. So a concentration gradient describes a difference in concentration between two regions. So let's take an example of this tea bag here. Once you put the tea bag in a cup of hot water, the tea is more highly concentrated near the tea bag than the rest of the cup. This creates a concentration gradient, right? There's a difference in concentration over here compared to over here. And we can actually draw an arrow because gradients are directional. So this gradient points down we say down the concentration gradient because it's going from high concentration to low concentration. So we can say the tea is going down its concentration gradient. It's going from high concentration to low concentration. And that's happening on its own. After time passes, the concentration of tea will be uniform throughout the cup, and there is no more gradient. So let's think about this again. The solute here are the tea particles, the solvent is hot water, and the tea molecules are going to go from where they're in high concentration, near the back, to where they're in low concentration, in the hot water. Over time, though, equilibrium, that's an important word, equilibrium will be reached where the tea particles are evenly dispersed with the water molecules. So there's no net movement in either direction. So this dissipation occurs by random motion. Of course, you can stir the tea if you want it to happen a little faster, but even just by leaving this out overnight, it'll reach equilibrium. So now we could talk about two different types of transport across a cell membrane. There's either passive transport or active transport. Passive transport is when you move down the concentration gradient, when you're moving something from high concentration to low concentration. And when you're moving down the concentration gradient, no energy is required for that type of transport. So again, passive transport is when you go from high to low concentration, no energy is needed. When you look at, when I write these brackets, that means concentration of. So this means you're going from high concentration to low concentration. Right, that's an example of like this ball rolling down the hill. It's rolling down the mountain. It's going from high concentration to low concentration, and no energy is required at all. That's going to happen on its own. Just by giving the ball a little nudge, it will roll down the hill on its own. And that's the same thing. It's passive. It does it without any energy required. So compare that to active transport. Active transport is when you move against the concentration gradient. So this is when you go from low concentration to high concentration. And when you go against the concentration gradient, that requires energy. So that's the equivalent of moving a ball up a hill, right? When you go from low to high, that's going to require a lot of energy, a lot of pushing. So we're going to see that substances get across the membrane either passively, if they're moving from high concentration to low, meaning there's more of it on the outside of the membrane than the inside of the membrane, then it can enter passively. However, if there's already a lot of the stuff inside of the cell and you're trying to get more of it in, you're going to need some energy. So that's active transport. So that should be clear before you move forward. The first type of passive transport we'll talk about is the simplest. Sometimes it's even called simple diffusion. We'll just call it diffusion. So diffusion is a type of passive transport because no energy is required. And this is the simple movement of molecules down a concentration gradient. For example, when you spray Lysol, the molecules are first concentrated in one corner where you spray it, but over time, all the Lysol particles will evenly diffuse through the room on their own, right? Diffuse is the key word, diffusion. They'll diffuse through the room and it will reach equilibrium. So no part of the room would smell any more strongly than another part of the room once equilibrium is reached. So diffusion is a type of passive transport. So again, fill in the blank. When blank is reached, the solute concentration is uniform and there is no more gradient. Right? Equilibrium is that word. So what affects the rate of diffusion? Right? There are a few things that affect the rate at which molecules can go from high concentration to low concentration. So temperature is one. So for example, on a hot day, garbage tends to smell a lot worse. It's not that the garbage is smellier when it's hot out. It just means that the air molecules, the air is hotter. So the odor molecules are going to be able to move and vibrate more rapidly. And they're going to reach your nose a lot more easily. Then when it's cold out, those odor molecules are not going to move around as much. They're not going to be able to diffuse from the garbage to your nose as easily. So on a hot day, there's more diffusion, so you smell garbage more rapidly. Also, air pressure affects diffusion and how fast molecules can move. And very importantly, the size of the molecule. So the smaller the molecule, the faster it can diffuse from point A to point B. right? So smaller molecules can dissolve faster and more easily. So let's look at this example. Here is a crystal dye that's placed in this fish tank. I don't like this example because it looks like it's harming the fish. But to illustrate diffusion, at least, you're going to see that at first, all of the crystal dye molecules are organized. They're all in a specific structure, right? They're in a crystal structure. But over time, as time passes, the dye molecules are going to diffuse with the water. So the dye molecules are going to go from where they're in high concentration to where they're in low concentration. The water molecules are going to go from high concentration to low concentration until there is no more net movement, right? Here there's equilibrium. There's an equal distribution of water and dye molecules. So there's no movement one way or the other. So that's an example of passive transport in diffusion. And is this dye hydrophobic or hydrophilic? Right, it has to be hydrophilic because it's dissolving in the water. Otherwise, it would be like droplets on the top, like oil. Another example of diffusion is found in gas exchange in the lungs. So every time we breathe in oxygen, inside these alveoli, which are the parts of the lungs that have the air inside, there's a lot more oxygen inside than in your blood. So we inhale oxygen, there's a lot more inside of our lungs than in our bloodstream. So since oxygen is a nonpolar molecule when it's in the gas form, so O2 is nonpolar, it's a gas. It can easily cross through the membrane. So this happens by passive transport. Since there's a lot more oxygen in the lungs than inside of the blood, the oxygen is going to be able to go rather quickly from high concentration to low concentration without using up any energy. So that's an example of passive transport in the human body. So gas exchange uses passive transport because you're going from high concentration to low concentration. And since oxygen is nonpolar, it can do so very easily. It can cross the membrane by itself. Right, but only small nonpolar molecules can cross the membrane passively by diffusion. And it's because that they're small and nonpolar that they can get through the hydrophobic barrier of the phospholipid bilayer. Right, this is not possible for large or polar molecules. If water is being transported from high concentration to low concentration, we call that osmosis. So osmosis is diffusion that refers to the solvent, meaning the water in our case, rather than the solute moving. So just like a solute, water moves from high concentration to low concentration, meaning water is going to always move to where it's more concentrated in solute. So let me try to rephrase that. Water is going to always try to dilute any salt. So if there's a region that's very salty, water is going to try to get there to try to make it less salty. So this is a special kind of tube. It's called a U-tube. Ironically, this is invented before U-tube was out. But it's shaped like a U, and on the bottom, there's a membrane that's selectively permeable. So in this case, it's a membrane that has tiny little holes in it. So only water molecules can get through. This solute, this sugar, cannot get through. It's too big. So what do you think will happen? Let's think before you don't look at this picture yet. Here we have two sides of the U-tube, and on one side there's a lot more solute. So there's a lot more sugar on the right side than the left side. And remember, what we would normally think is that, okay, so these sugar molecules will go from high concentration to low concentration, right, by diffusion. But there's this membrane here, and those solute molecules can't get through the holes of the membrane. But what will happen is water is small enough to get through. So where is the water going to travel? In which direction? Right, at equilibrium, all the water is going to want to try to go on the right side to balance out the saltiness. So what you're going to see at equilibrium is that both sides will have an equal concentration of solute because water went from high concentration to low concentration. So the water moves in such a way to make both sides of the membrane balanced in their solute concentration. I'm going to illustrate this one more time. This is called a Thistle Tube. And this is a type of tube that has a colored liquid inside. We'll say it's a 10% dye. And on the bottom of that tube, there's a membrane. It's a membrane that only allows water in and out. So let's say I have a 10% dye solution inside my Thistle Tube, and I put it in a 5% dye solution in the beaker. Pause the video here and tell me what's going to happen to the water inside this Thistle Tube. Is the dye going to leave into here? What's going to happen? Right? Remember that this membrane is only permeable to water. So that's my one hint. So pause here. And let's see what happens. What we can see is that after time, all of the water wanted to try to balance out the high concentration of dye in the Thistle Tube. So the water is going from the beaker inside the Thistle Tube to try to get it as close to equilibrium as possible. So after time, the concentration of dye inside the Thistle Tube will be less than 10%. And there's going to be more dye concentration on the inside as a result, right, of the beaker. Another way to think about this, I don't know if this is going to help or not, but think about the concentration of water, right? So what's the concentration of water inside this Thistle Tube? If it's 10% dye, that means it's 90% water. And what's the concentration of water inside the beaker? Right, 95%. It's 95% water inside the beaker because it's 5% dye. So here in the example, it's osmosis. Water's going from high concentration, 95%, to low concentration. Right, so that's where that water is going from high concentration to low concentration, and that's what explains the increase in volume of the Thistle Tube. So if this happens in an actual cell, the cell can change shape, right? So if you get an increase in water inside of the cell, it's going to change its shape. If you lose water from the cell, it's going to change its shape. So let's talk about how external salt concentration can affect the shape of the cell. In an isotonic solution, that means the salt concentration inside the cell and outside of the cell are the same. Iso means the same. Tonic, tonicity means refers to solute concentration, like the saltiness, so to speak, of a solution. So tonicity refers to how much solute is dissolved in solution, right? How salty is the solution. And an isotonic solution means that the cell placed in that solution has the same salt concentration as the solution itself, meaning there's an equal exchange of water in and out of the cell, right? It's already in equilibrium, essentially, so the cell shape won't be affected. So we like isotonic. And in the case of red blood cells, that's a 0.9% salt solution. So if you were to take red blood cells and put them in a 0.9% salt solution, they would not change shape. However, if you put them in a regular water solution, like just neutral water, if you put red blood cells in tap water, which has 0% salt, that means the solution would be hypotonic. There's less salt in the water than inside of the cell. What do you think would happen if a cell were placed in a hypotonic solution? So what do you think will happen if I put a red blood cell in tap water? Right, so since there's more salt inside the red blood cell, the water on the outside is going to try to balance it out. So what's going to happen? Will cells placed in a hypotonic solution swell, or will they shrink? So you could pause here if you need more time. But what will happen is these cells will swell up because the water is going to try to balance out the salt on the inside of the cells. And as they fill up with water, they might actually burst. So this is called hemolysis in the case of red blood cells. So cytolysis means cyto is cell, lysis is breaking. So cytolysis means the bursting of cells. And in the case of a red blood cell, we would call that hemolysis. So it's very important that blood cells are placed in the right concentration of salt, 0.9%, so they remain their proper shape. In a hypertonic solution, that means that the solute is saltier on the outside, right? The solution is saltier on the outside than the inside of the cell. And that means that cells will shrink. Cells will shrink because the water on the inside will try to balance out the high salt on the outside. So all of the water is going to leave the cell in a hypertonic solution, and that will cause the cell to shrink. And we can say that this red blood cell is crenated. Crenation is the shrinking of red blood cells. And that happens in hypertonic solutions. Right? In isotonic solution, there's no difference in solute concentration, so the cell remains its nice shape. If the cell is placed in a hypotonic solution, water will want to flow from high concentration to low concentration, and that will swell the cell. So plants really rely on hypotonic surroundings to make sure that they have the right water content and the right turgor pressure. So plants usually like to keep their solute concentration higher inside the cells than outside. So water is constantly going inside of the cells, right? The water is going to always want to try to balance out that the high salt concentration inside of the cells. A hypertonic surrounding results in the loss of water. So if you put salt water around the cells, what's going to happen is all the water from the vacuoles will try to balance out the salt on the outside. So the water from the vacuoles will be lost to the outside and the cells will shrink. And that will cause like the wilting of this plant. So salt kills plants because it draws out all the water from them. So plants prefer a hypotonic surrounding. So this is a good overview of how salt affects both animal cells in hypotonic and hypertonic solutions, and then you see plant cells. And you'll see something very interesting. So as the cytoplasm shrinks, you'll see all the chloroplasts, all these green organelles kind of clump up in the middle. And you can see that under a microscope, and you'll see that in a lab, hopefully. You'll be able to see how these different cells change shape in isotonic, hypertonic, and hypotonic solutions. So just a little fun fact, a marine turtle cries hypertonic tears after it lays eggs on land to get rid of any excess salt. So all organisms have to maintain this salt balance, right? It's part of homeostasis. So this marine turtle that was just underwater is now on land and has to balance the salt inside of its body. So it's going to cry to just get rid of excess salt. Interestingly, butterflies require a lot of salt in their diet, and they don't really have a way to get salt in their normal food. So what some butterflies do is they could drink the turtle's tears to maintain homeostasis. So this is, again, a part of like this circle of life, how all organisms rely on each other. So I'm going to let you look at this video on your own. You could click on this link. But right now you can see this video of a marine turtle that is secreting hypertonic tears from its eyes, and the butterflies are drinking the tears in order to get salt for their diet. So all the organisms rely on a certain concentration of salt for proper functioning. So again, here's the link below. You can watch that on your own. So that is the end of diffusion and osmosis. Let's talk about another type of passive transport called facilitated transport. So facilitated transport is a variation of passive transport because no energy is needed, because molecules go from high concentration to low concentration. So we said as long as molecules are going down the concentration gradient, we call that passive transport and no energy is required. What's different about facilitated transport is that this is the transport of polar molecules. So these are molecules that can't just diffuse across the membrane because they can't get through the phospholipid bilayer. So in facilitated transport, we have polar molecules like glucose, sugar, or amino acids that need to cross the membrane, and they do so with the help of carrier proteins. So these are special membrane proteins that allow the polar molecules to go down the concentration gradient. Right, so in facilitated transport, we see polar molecules move across the membrane by carrier proteins from high concentration to low concentration. Sometimes this is called facilitated diffusion because it's the same thing as diffusion, except it's facilitated by a membrane protein. So you can see at first we have a lot of polar solute that needs to get from high concentration to low concentration. So one by one, they will be able to bind to this carrier protein, and the carrier protein can change its conformation to allow the solute inside the cell one by one. So there's a conformational change, and this happens without any energy because you're going from high concentration to low concentration. So there's a video in the ebook that I suggest you watch. It talks a little bit about facilitated transport. Here you could just see how one molecule at a time is being transported. If you want the narration, you can find that within the ebook. All the videos are found on Connect. So that's the end of passive transport. What if we need to transport molecules against the concentration gradient? Right? What if we need to go from low concentration to high concentration? That's what active transport is for. So active transport needs ATP for energy, right? It's active, so we need ATP. You can remember ATP for active, A and A. And not only do we need energy, but we still need help from specific carrier proteins. So active transport is when we already have tons of something inside of the cell, but we still need to put even more in. So we're going from low concentration to high concentration. So we need ATP for that. And we also need a special protein on the membrane. The main example of active transport that you must know is the sodium-potassium pump. So the cells in your body, especially your neurons and muscle cells, must have a low concentration of sodium and a high concentration of potassium relative to the outside of the cell. Make sure that's in your notes. All right, so you want to make sure you always have more sodium on the outside than the inside, and more potassium on the inside than the outside. And so again, you would imagine that these ions want to always balance out, right? Salt is going to always want to try to get inside of the cell, right? Sodium is always going to try to get inside to try to balance it out. So we need the sodium-potassium pump to constantly get rid of sodium, to pump it out, and to constantly get more potassium in. Right, just like a pump of water, right? You're getting water from low concentration to high concentration. You need to actively put in energy for a pump. So the sodium-potassium pump uses ATP for energy to move sodium ions out of the cell and potassium ions into the cell, both against their concentration gradients. So the sodium-potassium pump is an example of active transport. We can see here. Okay, so step one is ATP will bind to the transport protein along with 3 different sodiums from the cytoplasm. So 3 sodiums can exit the cell. What will happen is the whole sodium-potassium pump will change its shape to then allow potassiums inside of the cell. So the whole protein changes shape. 2 potassiums could then enter the cell. And then again, that could increase the concentration of potassium inside the cytoplasm. So overall, and I'll show you a video for this, every time we use 1 ATP, we're getting 3 sodiums out and 2 potassiums in. So why does a sodium-potassium pump require energy? So pause here. Right, and the answer is C. So because it moves solutes against the concentration gradients, that's why energy is required. It's not because ions are being transported, and it's not because of membrane protein. Right? The reason why it's active transport and requires energy is because we're transporting solutes against their concentration gradient. We're going from low concentration to high concentration. So again, this video is in your Connect website. It's in the ebook. And this shows you how the 3 sodiums get kicked out and the 2 potassiums are allowed in with every ATP. So cystic fibrosis is a disease that results from a defect in active transport. So cells must maintain a very, very, very low concentration of chloride inside the cytoplasm. And cystic fibrosis is a result of a faulty chloride channel protein called CFTR. So normally there's this protein on the membrane called CFTR that moves chloride ions to the outside of the cell, actively transports them outside of the cell to make sure the inside does not have a lot of chloride. If you have a mutation in this CFTR channel protein, it cannot move the chloride ions anymore. And that causes a sticky mucus to build up on the outside of the cell, which could block airways and can clog the pancreas. So here you can see normal lungs, you don't see any mucus, but all this opacity over here is the mucus that's building up outside of the cells that can't transport their chloride ions out. So that's cystic fibrosis. So it's a genetic disorder because you inherit the mutation that doesn't allow this channel to take the right shape, right? So if the structure is disrupted, the function will be disrupted. So you can take a look and pause here. I wanted to just point out specific mutations. So what does this mean? What do you think F508del means? What do you think G51551D means? These are different mutations in cystic fibrosis protein, in the CFTR protein. Right, so what is G551D? What can that possibly mean? So if you remember, all proteins are made of amino acids, and all amino acids have a 1-letter code. So what this means, what G551D meant, was that normally the 551st amino acid in CFTR protein was a glycine. But instead, there was a mutation that caused it to be substituted for a D, for an aspartic acid. So that's a common mutation in cystic fibrosis, that you can have this kind of mutation in your DNA. And if you have that DNA mutation, that causes the ribosomes to make a mistake when they put together the amino acids. And if they can't form the proper shape, then they can't perform the proper function. So now we're going to talk about bulk transport across the membrane. So now we're finished talking about active transport. We'll talk about bulk transport. So large molecules are transported into or out of the cell inside vesicles via bulk transport. So remember, vesicles are also made of phospholipids. They're like those little bubbles of phospholipids. And if we're trying to get cells to secrete materials out, right, we're trying to get stuff outside of the cells, we call that exocytosis. Exo means outside. So when we get materials outside of the cell, we call that exocytosis. If we're trying to get stuff inside of the cell, we call that endocytosis. Right? If the cell eats materials from the outside, that's endocytosis. And regardless whether it's exo or endocytosis, that's called bulk transport. And bulk transport requires motor proteins to carry these vesicles along the cytoskeleton using energy from ATP. So take a second to pause here. Think about what motor proteins would carry vesicles and along which type of cytoskeletal protein. Right, so we spoke about that in the last chapter. Right, that microtubules are these tracks, and they get these motor proteins like kinesin can carry vesicles and walk along microtubules to go from point A to point B within a cell. In the case of exocytosis, point A is inside of the cell and point B is outside of the cell. So in exocytosis, vesicles surround the substance that needs to be exported outside of the cell. So these substances must be very large or polar, so they can't cross on their own. So they need to be packaged in membrane, in vesicles. And then the next step is they move to the cell membrane and they merge with the membrane, right? Remember, it's made of phospholipids as well. So because the vesicle is made of phospholipids and the cell membrane is made of phospholipids, they can easily merge and fuse. And once that happens, all of these large particles can be released to the outside of the cell. And this is regardless of the gradient. So whether it's high concentration or low concentration, the substance is so large that it's going to require vesicles regardless. And endocytosis is the reverse. So a small portion of the membrane will then bud inward, kind of trapping these large particles from the outside, and a vesicle will be formed, which can bring the particles into the cell, and the vesicle will then unwrap and then allow all the particles inside of the cell. So endocytosis comes in 3 varieties. So they can internalize materials from the outside in one of 3 ways. The one is called phagocytosis. Phagocytosis is when large solid material is taken in by endocytosis. So when your macrophages, right, phage like phagocytosis, your macrophages are types of white blood cells that can literally engulf viruses and gobble them up and internalize them. So phagocytosis is like cell eating. You're engulfing large particles. Pinocytosis is like cell drinking. So little vesicles form around a liquid and kind of internalize very small little droplets. So that's endocytosis still, because you're internalizing, but it's not large particles that are getting inside. They're very small particles. We call that cell drinking. And finally, you can have receptor-mediated endocytosis, which is the most complicated of them all. And that's when vesicles are coated with a special type of receptor protein. And that tells the vesicle where to go. So it can the vesicle could be sent to a specific cell location based on the type of proteins that are outside of the vesicle. And I'll explain that in just a bit. So here is phagocytosis. This is engulfing a large particle, getting it inside of the cell. This is pinocytosis, right? Cell drinking. We're getting some smaller molecules inside of the cell by a vesicle. And here we can see receptor-mediated endocytosis. So here there are actual membrane protein receptors on the outside of the cell waiting for the solute to bind. And once enough solute can bind, this whole thing starts to bud inward and form what's called a coated pit. Once there's a coated pit, right, once it's coated with solute, the receptors signal to each other and this whole vesicle pinches off. And this whole thing could then go inside of the cell and unwrap. So this solute can be released inside of the cell. So those are the 3 methods of endocytosis. And that can be reviewed on your own. Again, this is one of the videos in the ebook. Right there you can see receptor-mediated endocytosis. So pause here and answer this question. So here we can see molecules are going from high concentration to low concentration, but it requires a membrane protein. So the answer is A. So it's important to keep track of these different types of transport. You should know for each type, are you going from high concentration to low concentration or vice versa? what's needed. Do you need a protein? Do you need ATP? And what's an example? So here's my little cheat sheet, but the book also has an example in table 5.1. So for each type of transport, which direction are you going in? What's required? What's an example? And again, here's another table for you to review. So I want to end off by talking about what's outside of the cell membrane. We spoke about a lot what's inside the cell membrane, but what's outside of a cell membrane? The extracellular matrix is what's outside of the membrane. So here is the cytoplasm on the inside, and outside is the extracellular matrix. And the extracellular matrix varies greatly depending on different types of cells. So depending if you're like a bone cell or a blood cell or a hair cell, you're going to have a very different type of extracellular matrix. But regardless, think of it as a meshwork of proteins and sugars and other polysaccharides that are just tangled with it. It's like an entangled mesh of sugars and proteins. And they're in close contact with each other. Some of the proteins found in the extracellular matrix include collagen. So collagen is a very important protein that maintains structure and shape in all of our tissues, especially muscle and our skin. So collagen resists stretching. So here's a collagen fibril over here. Also, elastin is another type of extracellular matrix protein that helps provide resilience and more strength. There's also fibronectin, which integrate with integrins. So fibronectin and integrins kind of work together to make sure that the cell is anchored properly. So what happens is this integrin protein can connect the cytoskeleton inside of a cell to the extracellular matrix. So it's like a staple in a way. It's keeping the cell in place from the inside out. And over here you can see proteoglycans. Looks like a spider web type thing, like its branch structures. These are combinations of sugars and proteins. That's what makes up most of the gel outside of the cell. Like the extracellular matrix, a lot of proteoglycans. And this again helps resist any compression of the extracellular matrix. So outside of the cell, we have like a gel of proteins and sugars. But again, it varies greatly from cell to cell. And again, depending on what type of cell you are, you might be associated with each other very tightly. So like skin cells are types of epithelial tissue. Epithelial tissue have very tightly attached cells. There's no space in between the cells. That's very different than a red blood cell, which is floating on its own. But cells are rarely isolated from each other, right? A red blood cell is an example of a cell that's isolated from other cells pretty much. But many cells require physical contact with each other or with the extracellular matrix in order for it to function properly. So there are three main types of cell surface junctions in animal cells that help cells connect to each other or to the extracellular matrix. We'll talk first about adhesion junctions, then tight junctions, and finally gap junctions. Adhesion junctions help animal cells stick together. So adhesion junctions are like rivets that mechanically attach adjacent cells, almost like a button, a snap button. We're attaching these two adjacent cells together. And the adhesion junction proteins connect cells to other cells and to the extracellular matrix outside of the cell. So this is pretty unique. These junction proteins, so this is the extracellular matrix where I'm pointing right now. So these proteins go through the extracellular matrix and connect the cytoskeleton of one cell to the cytoskeleton of another cell. So this is a very strong connection. It's like reaching in from bone to bone. An example would be a desmosome. This is shown as a desmosome. Desmosomes link intermediate filaments from one cell to intermediate filaments of another cell. So this is called an adhesion junction. Sometimes they're called anchoring junctions because they anchor adjacent cells together. So it's very important for strength, very important for epithelial cells. The next type of junction is called a gap junction, and this helps cells to communicate very rapidly. So in gap junctions, proteins form this kind of a tunnel through the membranes of two neighboring cells. So there's 6 proteins that come to form a tunnel, and molecules can go very rapidly from one cell to the next. They don't have to rely on exocytosis and endocytosis to go from one cell to the next. They can just go through these little secret tunnels made of proteins. So the membrane proteins kind of line up in a certain way to form a channel for small molecules to go through. So these can be ions or very small molecules. Finally, we have tight junctions. And in tight junctions, certain proteins fuse the membranes of adjacent animal cells together. So here is cell A and here is cell B, and the membranes are fused together by groups of tight junctions, almost like stitches, like sewing machine stitches. And this allows sheets of cells to form a leak-proof barrier between them. So this is important in the intestines, where you have like epithelial lining. We want to make sure all of those cells are very tightly attached and form a waterproof seal. Right? We don't want any of the food we digest to get inside of our tissue. So we need this leak-proof barrier between our cells. And plant cells don't have these types of junctions. They don't have the same types of connections. But what they do have is something called plasmodesmata, which are equivalent to gap junctions. So gap junctions in animal cells are analogous to plasmodesmata in plant cells. And these are these little spaces in between the cell walls that allow the passage of materials. So it's important, those cell walls are very thick, they're made of cellulose, so there needs to be little gaps to allow materials to go and go from one cell to the next. So those are plasmodesmata. So that is the end of chapter 5. You also have some more coming up. We'll talk about enzymes and energy next. But just to review the membrane proteins, the different types, we can see we have adhesion junctions, which attach one cell cytoskeleton to the other cell cytoskeleton. Then we had tight junctions, which form that leak-proof barrier. And then here we have the gap junctions. Right? And here again, tight junctions, adhesion junctions, gap junctions. So that's the end, and I hope that wasn't too boring. I know it's not the most exciting stuff to learn about, but you'll see that in biology, you need to understand the fundamentals of just how the cell works before you can learn the really interesting and fascinating stuff. So hang in there, the fun stuff is about to start.